Dear students, today we will talk about medical embryology, and namely we will talk about implantation, development of placenta and other extraembryonic organs. So here we can see that during the embryonic development there is a development of embryo, but embryo during intrauterine period of life cannot exist without extraembryonic organs. They are placenta, chorion, amnion and others, which provide development of embryo. They provide connection between embryo or fetus with mother's structures, and also they provide nutrition and protection for the developing embryo or fetus. So here we can see embryo or fetus and surrounding structures which form extraembryonic organs, and today we'll talk about them. So let's revise the stages of human embryogenesis. Human embryogenesis is divided into early and late stages. Early stages, they include fertilization, which results in formation of zygote. Next, it's cleavage. It takes first week and it results in the formation morula and then blastula. So blastula is the final result of cleavage. After that, there is gastrulation, which includes early and later gastrulation, and it results in formation of 3 embryonic layers. Next stage is axial organs development, and it results in the formation of axial organs. After that, formation of body of embryo. It results in the formation of body of embryo, and gastrulation takes place at the same time with axial organs development and formation of body of embryo, namely late gastrulation. And also there are late stages of embryogenesis. They include histogenesis and organogenesis. So it's development of tissues and organs and systems of organs. And early stages of human embryogenesis, they are discussed in other topics, in other videos, and today we will talk about the development of the extraembryonic organs, how tissues and organs do provide development of embryo, its nutrition, feeding during intrauterine development. So, embryogenesis begins from fertilization, and there is a period of progenesis, which includes ovogenesis, formation of ova, female gametes, and it takes place in the follicles of ovaries. And also there is spermatogenesis, process of formation of spermatozoa, male gametes, which are developed in the testes in spermatogenic epithelium of convoluted seminiferous tubules. And after that they meet, and the result it's fertilization, first stage of the embryogenesis. So there is a preliminary period which is called progenesis, which includes ovo and spermatogenesis. And here we can see ovary with different follicles, and its mature follicle or Graafian follicle, its fallopian tube, and in the middle of the ovarian cycle, this follicle undergoes ovulation. When high level of estrogens activates release of LH, luteinizing hormone, ovulatory quota of LH activates ovulation, and ovulation begins. There is ischemia of the wall of ovary, and after that there is necrosis. This wall is broken, and also wall of the follicle is broken, and ovum is released from the follicle, and it goes to the fallopian tube, and there is ovum surrounded with corona radiata. And here in ampulla of fallopian tube, it's waiting for the fertilization. And here spermatozoa are coming, and it's contact phase of fertilization. There is distant phase before their meeting, and it's contact phase. And if fertilization occurs, it results in the formation of zygote, unicellular embryo, and it goes from the fallopian tube to the cavity of uterus. And here we can see its ovary. There are 2 ovaries, 2 fallopian or uterine tubes or oviducts, and after the ovulation, ovum, or now it's secondary oocyte. Before it was primary oocyte in the follicles, now it's secondary oocyte, and it's released in the ampulla of fallopian tube. And spermatozoa are coming here, and fertilization occurs. So distant phase when spermatozoa are coming through the female reproductive tract, through vagina, uterus, and they are going to fallopian tube. They receive signal from the ovum because it releases hinogamons. They are chemoattractants. They attract spermatozoa, and they are moving to the place of fertilization. And after the fertilization, zygote, it undergoes development. There are following stages. It's cleavage, And during the cleavage, zygote migrates from the place of fertilization, from ampulla of fallopian tube to the cavity of uterus. It takes about 5 days, and till 7th day it's already in uterus, and here it undergoes attachment to the wall of uterus, and it's called implantation. So here we can see follicular cells which surround ovum, or cells of corona radiata, and cilia or of epithelium of fallopian tubes. They move this complex of cells to the place of following fertilization. And here we can see ovum, which is waiting for the fertilization, and spermatozoa are coming here. It's distant phase, and also muscle contractions, they help in this. Thermotaxis, when they are going to the normal temperature to the higher temperature. Also rheotaxis, its ability of spermatozoa to move against fluid flow. And chemotaxis, they are moving to the ovum, which releases henogamons, and they attract spermatozoa. So there are factors which help in the distant phase. And also distant phase, it includes capacitation, its final maturation of spermatozoa, when components of the glycocalyxes are removed from the surface of spermatozoon, and spermatozoa, they become to be able for the fertilization. So its distant phase, it includes positive rheotaxis, positive thermo and chemotaxis, and also it includes capacitation of spermatozoa. And when they achieve the ovum in the fallopian tube, contact phase begins, and it includes acrosomic reaction when spermatozoon releases enzymes from the acrosome. Also, it includes a cortical reaction when ovum releases cortical granules, and also zonal reaction when zona pellucida becomes to be unpermeable for another spermatozoa. And result of fertilization, it's formation of zygote, and zygote begins its cleavage and migrates from the fallopian tube to the cavity of uterus. And here we can see zygote, it's result of fertilization. Here we can see 2 pronuclei, female and male. There are 3 polar bodies, which are result of 2 meiotic divisions. And its zona pellucida, now it's unpermeable for another spermatozoa. Follicular cells, or cells of corona radiata, they are lost, and this process is called denudation, nude, because those cells aren't needed anymore, and zygote exists without corona radiata, so only zona pellucida remains. And here we can see cytoplasm of zygote. It contains yolk granules which were received from ovum, and those yolk granules are needed for the nutrition of embryo during first week till the implantation. Here we can see zygote, 2 pronuclei, it's cell membrane of zygote, it was membrane of the ovum, and now it's membrane of zygote, and it's zona pellucida. So it's called zygote, unicellular embryo. And the next stage of embryonic development, which takes 1 week, it's cleavage. It's mitotic divisions of zygote, and cells which are developed from zygote are called blastomeres. Here we can see stage of 2 blastomeres, stage of 3 blastomeres, 4, 5, and the following divisions. Here we can see stage of morula. It's nodule of cells of blastomeres. Here we can see morula. And those cells, they begin to produce fluid which fills the space between them, so cavity develops, and size of cells decreases, and now this embryo is called blastula or blastocyst. Before it was morula, cells were smaller and their number was smaller. Cells were bigger and number of cells was smaller, and now number of cell increases, size of cell decreases, and also cavity appears. And it's difference between morula and blastula. Blastula, it has cavity, cells are more numerous, but they are smaller than morula. And feature of cleavage, which differs it from usual mitotic divisions, its absence of interface, and cells they aren't growing between the divisions, and cells become to be smaller and smaller. Size of each individual cell becomes to be smaller and smaller, but their common volume is the same because entire volume is fixed. There is zona pellucida, and cells they don't grow, so common volume remains the same, but each individual cell becomes to be smaller because number of cell increases. So finally, till fifth day, embryo is called blastula or blastocyst. Blastocyst is kind of blastula which is characteristic for human and in other placental animals, those animals which have placenta. And there are in other kinds of blastula, amphiblastula, holoblastula. That's why blastula is more common term than blastocyst, and blastocyst is individual kind of blastula which is characteristic for the placental animals. So here we can see blastocyst or blastula, and now it migrates from fallopian tube to the cavity of uterus, and it achieves cavity of uterus on the fifth, sometimes fourth day, and after that it's looking for the best place for the implantation. So blastula or blastocyst, name of blastula in human, it has two groups of cells. There are outer cells which form the wall of blastula, and also inner cells, nodule of cells. So blastocyst, it has embryoblast. Those cells which are shown in green on this picture, they will form embryo, only these cells, and also they will form some extraembryonic organs. And these cells which form the wall of blastula, they are called trophoblast. So trophos, it means nutrition or feeding. So we make a conclusion that trophoblast will provide nutrition or feeding of embryo, and embryoblast will form embryo, and also it will form some extra embryonic organs. So here we can see two basic parts of blastocyst. And now let's look what is the further development of those structures and parts. Here we can see microphotograph of blastocyst. It's embryoblast, it's trophoblast, it's blastocell cavity of blastula, and it's zona pellucida. It's the structure of blastula. And during this cleavage, embryo, zygote, then morula, it migrates through the fallopian tube and achieves the cavity of uterus on the fourth day. Now it's a free period of life of blastula. Previous period it was tubular period, and now it's free period when it's freely located in the cavity of uterus. And till seventh day it will choose the best place for its attachment, and it will undergo implantation, attachment to the wall of uterus. So this period it takes 1 week, and we'll see that uterine tube, it's oviduct, duct for the embryo. And when oviduct has some constrictions, some problems, sometimes embryo cannot pass through the fallopian tube and it cannot achieve the cavity of uterus. So it will get implanted in the fallopian tube, and it will be called extrauterine pregnancy. It's complication, and it requires surgical removal of the fallopian tube. So in normal condition, it achieves the cavity of uterus, and it looks for the best place and undergoes implantation. But let's look at the blastocyst. Here we can see that it's located inside zona pellucida. Here we can see, and it prevents its implantation through the passing through the fallopian tube. So it prevents implantation in the fallopian tube, and also it keeps all blastomeres together to not be separated, and also it prevents polyspermia during the fertilization. In other spermatozoa, after the fertilization by one, they cannot penetrate zona pellucida, because also it can be called fertilization membrane. So 3 functions: it's prevention of polyspermia, it keeps blastomeres together, and it prevents implantation in the fallopian tube. But now, when it achieves the cavity of uterus, blastocyst should be somehow implanted. It should be attached to the wall of uterus, and this zona pellucida, it doesn't admit it. So what will happen to this? Next process which will happen, it is called hatching, like hatching of chicken. So here we can see blastocyst, it's zona pellucida, and before the implantation it's broken, and embryo goes away from the zona pellucida. It's released from zona pellucida, and it leaves zona pellucida, and now cells of trophoblast are freely exposed on the surface of blastula. They are able to undergo attachment to the wall of uterus and invasion to the wall of uterus, and this process is called hatching. This zona pellucida, it disappears, and this blastula or blastocyst, it continues its development. Here we can see blastocyst, and it goes away from zona pellucida, and we call this hatching. Here we can see blastocyst, it excites from zona pellucida. And here we can see blastocyst in zona pellucida inside, and it's zona pellucida, and it's blastocyst which leaves this zona pellucida. It's called hatching. And after the hatching, blastocyst is able to undergo implantation. It's looking for the best place in the uterus, and if in the uterus there are some scars, it looks for the better place. So sometimes if there are some diseases in uterus, it may cause infertility because blastocyst cannot be implanted because of diseases of endometrium. But in normal condition, it chooses the best place after the hatching and is attached to it. So the next process begins. It's called implantation. Implantation is the process of attachment to the wall of uterus, and it takes place usually on the 7th day. Sometimes you can find information in the textbooks from 6th or 5th day till 8th day. So it's term of the implantation, but the average is day 7. So here we can see blastocyst, it's embryoblast and it's trophoblast, and here we can see that those cells, they are like epithelial cells, and they are tightly attached to each other, and those cells, they will play key role in the implantation. They will release hydrolytic enzymes, and first of all they have receptors which are needed to the attachment to the endometrium. So receptors, they play very important role in the attachment, and also these cells, they will release hydrolytic enzymes which are needed for the invasion. And during the implantation, trophoblast is divided into 2 parts. These cells, which are distinct cells, so they form inner layer, they are called cytotrophoblast. Cyto because it's made up of distinct cells. And some cells of such trophoblast, they undergo some mitotic divisions, and they are pushed out from cytotrophoblast, and they are fused. So they form syncytium. They form projections alike villi. Here we can see it's villus, and it's cell mass, which is a result of fusion of cells of cytotrophoblast. Some cells, they migrate out from this layer, and they are fused, and their common cytoplasm exists with numerous nuclei, and it's called syncytium, and we call this layer of trophoblast syncytotrophoblast. So it appears on one place, on one point where blastocyst is attached to the uterus. It's the first villus formation, but later those villi and layer of syncytotrophoblast appears on entire perimeter of the blastocyst. So it's syncytotrophoblast outer layer. It's made up of syncytium, supracellular structure, which is result of fusion of cells. And here we can see or it's called symplastic trophoblast as well. In different textbooks you can find both names. It's a result of fusion of cells which are migrating from cytotrophoblast. And remember those two layers, cytotrophoblast and syncytiotrophoblast, because they play key role in the development of placenta, and they play key role in the nutrition of embryo during the embryonic development. So first phase of implantation is called adhesion. It's only attachment of blastocyst to the surface of endometrium. It's adhesion, first phase of implantation. And next phase is called invasion. When syncytiotrophoblast appears, it releases hydrolytic enzymes, and they dissolve wall of uterus, and embryo is embedded in the wall of uterus. It goes deeper, and it undergoes it completes implantation, so it goes to the functional layer of endometrium. So implantation, it has two phases. First is adhesion, simply attachment, and second is invasion. Embryo is implanted in the functional layer of endometrium. Here we can see functional layer, deeper it's basal layer, and those layer, functional layer, it's those layer which undergoes cyclic changes during the menstrual cycle, and also this layer, it provides implantation of embryo, it keeps embryo after the implantation, and it undergoes changes during pregnancy, it forms decidua. Later we will talk about this. And also it's involved in the formation of placenta. So functional layer, it's very important in the formation of extraembryonic organs and in the nutrition of embryo during the pregnancy. And here we can see endometrium. There is basal, it's functional layer. There are uterine glands. Here we can see they are coiled because they are under influence of progesterone. It's luteal phase, which follows the ovulation. So after the ovulation, there is luteal phase, and progesterone, it prepares uterus for the possible pregnancy. And here we can see different arteries which provide nutrition of these layers. There is arcuate artery, which is located in myometrium. It has radial branches, and here we can see that they are going to endometrium. And radial arteries, they have two kinds of branches. There are straight arteries, which provide nutrition of basal layer, and basal layer, it's not involved in the formation of placenta. And spiral arteries, they are going to the functional layer. So here we can see distinct source of blood supply of functional layer. It undergoes nutrition from the spiral arteries. And now let's look at this endometrium and what is going on during the implantation. Here we can see spiral glands. They are coiled because of secretion. They produce mucus, which is needed for the nutrition of embryo. And between them, in stroma, in connective tissue, there are decidual cells rich with glycogen, which also may be used by embryo for nutrition. Here we can see arcuate artery in myometrium, radial, straight artery, and spiral artery, which forms branches which are going to functional layer of endometrium. So blastocyst is coming here, and it looks for the best place for the implantation. And now it got attached to the wall of uterus, and we call this adhesion. It's the first stage of implantation. It's adhesion. And receptors on the surface of the cells of trophoblast, they play key role in this attachment. So these cells, their receptors, they choose the place for attachment, and here we can see that adhesion is completed. And after that, cells from cytotrophoblast, they migrate out and they form the villi of syncytiotrophoblast. They are fused, and syncytiotrophoblast, it releases hydrolytic enzymes which dissolve the wall of uterus, and it helps in the invasion when embryo is going inside the endometrium. And embryo goes inside, it undergoes invasion. Here we can see, and syncytiotrophoblast appears on entire surface of blastocyst. So here we can see completed stage of invasion, and it's completed implantation. and a place where embryo was invaded, this surface is covered by epithelium, so embryo completely enters in the functional layer, and it's covered with wall of functional layer of endometrium. And here embryo receives nutrients from mother's tissues, from tissues of the endometrium. First of all, it receives nutrients from the secret of uterine glands. Also, dissolved tissues of uterine wall, they also give nutrients to the embryo, and also the decidual cells in the tissues, they are rich with glycogen, and they also are used for nutrition. And this nutrition or feeding of embryo is called histotrophic. Histo, it means tissues, so embryo receives nutrients from tissues of the uterus. Here we can see this embryo, so syncytotrophoblast or symplastic it receives those nutrients, and the cells, they give those nutrients to the cells of cytotrophoblast, and they achieve cells of embryoblast. And also these villi, they keep growing, they produce hydrolytic enzymes, and till third week, so all this process, it takes second week, till third week, these villi, they achieve blood vessels of mother, blood vessels of functional layer, and blood vessels, they release blood, they are damaged, and blood flows away from blood vessels, and it forms cavities filled with blood, which are called lacuna. Here we can see lacuna and villi which are included into lacuna. And embryo begins to receive nutrients from mother's blood. Here we can see mother's blood. It contains different nutrients, and embryo receives nutrients from it, and nutrients are coming to the cells. And it begins on the third week, but histotrophic type of nutrition it's still going on. So now embryo has two types of nutrition, and till the second month, histotrophic type of nutrition is completely over, and hematotrophic type of nutrition is the predominant type of nutrition, and it is the main type of nutrition till the delivery, till the childbirth. So here we can see what is going on during the implantation and result of implantation. When embryo is completely located in the wall of uterus, and it achieves mother's blood vessels and receives nutrients from it. And here we can see first stage of implantation of invasion, when embryo dissolves tissues of mother's wall of endometrium. It's histotrophic type of feeding. Here we can see syncytotrophoblast, it's shown in green on this animation, and there are uterine glands. So here we can see that hydrolytic enzymes, they dissolve these tissues, and embryo receives nutrients from tissues, and it's histotrophic type of feeding. It's second week, and it continues during end of the second month. And beginning from the third week, there is a hematotrophic type of feeding when embryo achieves blood vessels, and blood from mother's blood vessels flows away, and it forms lacuna, which are filled with mother's blood. And now embryo begins to receive nutrients from mother's blood, and this type of feeding lasts for it continues till the birth. So let's make a summary of what is going on during these stages. Implantation is the 7th day of embryonic development, and it includes two phases. First, it's adhesion, second is invasion, and result is completion of the implantation. Embryo is completely located in the functional layer of endometrium. And feeding of embryo, first of all, during first week till 7th day, till implantation, nutrition is provided by yolk granules. So before the implantation, embryo receives nutrients from yolk granules. During second week, when embryo completes invasion, after the adhesion, during the invasion, embryo receives nutrients from the tissues. And also, beginning from third week till birth, there is hematotrophic type of feeding or nutrition, and it continues till birth. And here we can see stages of implantation and types of feeding of embryo. And here we can see it's embryoblast, cytotrophoblast, and let's look what happens to embryoblast. This embryoblast, we know that it forms embryonic structures, and also it's involved in the formation of some extraembryonic organs, such as amnion and particularly chorion, allantois. So here we can see common cell mass, it's called embryoblast, and it enters in the period of implantation. Here we can see this cell mass, and it's divided into two layers. It's epiblast and hypoblast, upper layer and lower layer. Embryoblast, it will form ectoderm, and hypoblast, it will form endoderm. After that, those layers, they keep growing, and they are enclosed into, they form two plates, and those plates later will be enclosed in two vesicles. It's amniotic vesicle and its yolk sac. Two vesicles which develop from epiblast, its amniotic vesicle, and yolk sac, it develops from hypoblast. And place of the attachment, it's called embryonic disc. Now it's two-layered, it contains epi and hypoblast. And after that, some cells from epi and hypoblast, especially from epiblast, they will form mesoderm. And also syncytiotrophoblast appears during the implantation. And here we can see formation of extraembryonic mesoderm and embryonic mesoderm. It fills space between ecto and endoderm here, and also it will surround amniotic vesicle and yolk sac. Here we can see formation of extraembryonic mesoderm and embryonic mesoderm. It will appear here. So here we can see blastocyst, and it has amniotic vesicle and yolk sac. Its amniotic vesicle and its yolk sac, and place of the attachment, it's called embryonic disc. So amniotic vesicle, it has embryonic ectoderm and extraembryonic ectoderm, and yolk sac, it has embryonic endoderm and extraembryonic endoderm. So embryonic ectoderm and embryonic endoderm, they are attached to each other, and they form embryonic disc. Here we can see embryonic disc, which has embryonic ectoderm and embryonic endoderm. And also we have extraembryonic ectoderm, it's top of the amniotic vesicle, and extraembryonic endoderm, it's bottom of the yolk sac. They aren't involved in the formation of embryo, but they participate in the formation of the extraembryonic organs, such as amnion, it's derivative of the amniotic vesicle, and yolk sac, it's a derivative of the extraembryonic endoderm, definitive yolk sac. So here we can see the following stage. Here we can see it's cytotrophoblast, syncytiotrophoblast. Here we can find amniotic vesicle, it's a yolk sac. So blastocyst, it grows inside, and we can see that entire size has been increased, and amniotic vesicle and yolk sac now are small. And we can see that they are surrounded by some tissue. It's extraembryonic mesoderm. Between embryonic ectoderm and embryonic endoderm, embryonic mesoderm develops between those two layers. And also some cells, they migrate and they form extraembryonic mesoderm, which fills all these spaces. Here we can see extraembryonic mesoderm. It fills the space between trophoblast and yolk sac, as well as between trophoblast and amniotic vesicle. And now this mesoderm, it has a cavity. Cavity arises here, and this extraembryonic mesoderm, it forms cavity, which is called extraembryonic coelom. Here we can see this cavity. So it separates extraembryonic mesoderm, which covers yolk sac, amniotic vesicle, and also those extraembryonic mesoderm which lines the wall of blastocyst. Here we can see it. And also there is connecting or body stalk, which also is formed by extraembryonic mesoderm. And this connective tissue or embryonic connective tissue of extraembryonic mesoderm, it will be involved in the formation of the umbilical cord. It will form connective tissue of the umbilical cord. So this connecting body stalk, it keeps embryo with some extraembryonic parts, and it attaches this to the wall of blastocyst. And here we can see extraembryonic mesoderm, and also we can find here cytotrophoblast and syncytiotrophoblast, 3 layers of the wall of blastocyst. Here we can see syncytiotrophoblast, cytotrophoblast, and extraembryonic mesoderm. So those 3 layers, they form structure which is called chorion. So chorion, it includes cyto and syncytiotrophoblast, so it includes trophoblast, and additional structure is extraembryonic mesoderm. So chorion equals to trophoblast plus extraembryonic mesoderm. Trophoblast, 2 layers of trophoblast, and extraembryonic mesoderm, they form chorion. Here we can see blastocyst. It has trophoblast and embryoblast, and trophoblast is divided into cyto and syncytiotrophoblast. Here we can see them, cyto and syncytiotrophoblast, and also additional layer, it's extraembryonic mesoderm, it appears here. And those 3 layers, they form chorion, very important structure which is involved in the embryonic development of placenta. Here we can see chorion. And also chorion, it leads to formation of placenta. And today we will talk about this, how placenta is formed from chorion. So chorion, it includes formation of villi. Here we can see those villi, which are growing inside the wall of uterus, and they provide nutrition of embryo. So they are like roots of plants. And there are some kinds of chorionic villi. And we divide them into 3 groups or 3 kinds. There are primary, secondary, and tertiary. So primary, they are the earliest, then they become to be secondary, and tertiary, they are mature villi, mature kind of villi. So first of all, primary villi develop. And let's look at the primary villas. Here we can see cells of cytotrophoblast, and they are surrounded by the cells of syncytiotrophoblast. And here we can see 2 layers. So primary villi, they have only 2 components. They are made up of trophoblast only. Next are secondary villi. So what has been changed? Extraembryonic mesoderm grows inside these villas, and it appears here. It forms core of the villas, and the result is formation of secondary villas. And finally, fetal blood vessels, which are already developed in the embryo, it will be later stage of embryogenesis, they are growing from embryo to the chorionic villi. They carry here erythrocytes, it's endothelium, it's wall of the embryonic blood vessel. So now this villus is called tertiary villus or mature villus. And blood of embryo is present here. It can receive nutrients from mother's blood through the wall of villus. So let's make a summary. Chorion, it has villi. Primary villus, it has 2 components. It's cytotrophoblast and syncytiotrophoblast. So 2 layers of trophoblast. In secondary villus, extraembryonic mesoderm grows inside the villus, and it's 3rd additional component. So secondary villus, it's primary villus plus extraembryonic mesoderm. And tertiary villus, it includes cytotrophoblast, syncytiotrophoblast, extraembryonic mesoderm, and blood vessels of embryo. So it's 4th additional component. So tertiary villus, it's equal to secondary villus plus blood vessels of embryo, and those blood vessels, they make villi mature, tertiary villi. And here we can see this villus. And here we can see that villus is located in the lacuna with mother's blood. It's functional, and it's basal layer of endometrium. It's spiral artery, and it carries blood to lacuna. And this villus is located here, and here we can see barrier between mother's and child's blood. Here we can see blood of child, blood of embryo, and it's blood of mother. And this barrier between them, it's placental barrier, and it includes all components of tertiary villi. They are wall of the capillary of blood vessel of embryo, its extraembryonic mesoderm, its cytotrophoblast and syncytiotrophoblast. So there are basic layers of the placentary barrier. And also extraembryonic mesoderm, it contains macrophage cells. They are called Hofbauer cells, which provide immune supervision here. So here we can see placenta barrier, and this barrier, it provides different immune conflicts between mother and child, because child's cells, they are foreign for mother. So mother's organism, especially immune system, may kill embryo during the development. So this barrier, it separates two bloods, babies and mothers, and they haven't direct contact. So this placenta barrier, it provides exchange of gases. When blood of baby comes here, it's venous, and it receives oxygen from mother's blood, and also different nutrients from mother are coming to blood of embryo. And blood of embryo, it gives away carbon dioxide. It goes to mother's blood, and also waste products of embryo metabolism, they are released from embryo, from blood vessels of embryo to mother's blood. So it's a place of exchange of those components, and this barrier, it provides this exchange. Which components and which structures, which substances can pass through this barrier? Formed elements of blood, they cannot pass through this barrier, and different substances, nutrients, they can pass through, and immunoglobulins, for example, immunoglobulins, they cannot pass through this barrier, but immunoglobulins G, they are the smallest, and they can pass through this barrier. So mother's immune substances, immunoglobulins G, they pass through this barrier and they achieve blood of embryo. So they provide immune protection for embryo during the embryonic development and during some time after birth, mainly during first 6 months till 1 year. Mother's immunoglobulins may be present in blood of embryo. So here we can see this placenta barrier. But also some viruses and some in other bacteria, also they can pass through this barrier, but usually this barrier is strong enough to prevent this. And here we can see that villi, they are growing deeper. Here we can see secondary villus formation, it's primary, secondary, it's formation of tertiary villus, and also they undergo branching, they are branched, they are attached to the endometrium, and they are branched, and they form something like tree branched structure. So it's formation of placenta. Here we can see chorion, and it includes syncytiotrophoblast, cytotrophoblast, and extraembryonic mesoderm. And here we can see that villi, they develop mainly on one pole of chorion, where it's attached to uterus, where it's growing inside the uterine wall, and where it forms placenta. So we have two parts of chorion. It's chorion frondosum with villi, and smooth chorion without villi, it's called chorion leve. And this part of chorion, only chorion frondosum, it will form placenta on this pole, and chorion level, it doesn't form placenta. So here we can see this villus. It is located in lacuna, and it will be attached to the endometrium here. It will be anchoring villus or stem villus, and it will form branches, and those branches, they will form branched tree-like structure or structure-alike root. And here we can see this branched structure. It's anchoring villus and its secondary branches, and this structure of placenta, it's called cotyledon. Cotyledon is lobule and structural unit of placenta. Here we can see villus, its stem or anchoring villus, which is attached to the endometrium, and also there are flatten villi, which aren't attached to the uterine structures. They are located in the maternal blood, and they receive nutrients from maternal blood, and it's called cotyledon. So structure like root of tree or like something like tree, and those roots they are located in the mother blood. So there are flatten villi and its anchoring villus. And its lacuna, cavity where cotyledon is present, its lacuna, and blood of mother, it comes here from spiral arteries. It circulates here, and venous blood, after blood exchange, after gases exchanges in the villi, venous blood goes to the venous vessels. And between the neighboring cotyledons there are septa. Here we can see septum, its projections of the endometrium, which separate neighboring cotyledons. And here we can see one cotyledon, there are villi of chorion, and another cotyledon, also villi of chorion. There are anchoring villi and flatten villi, it's villus tree. And between them, lacuna are separated by septa. Here we can see septum and its lacuna, cavity filled with blood. It's septum and its lacuna. So villus tree is located in own lacuna, and lacuna are separated by septa. And this cavity, lacuna with septa with villus tree, it's called cotyledon. And here we can see cotyledons. They may be bigger, they may be smaller, their number is different. And here we can see anchoring villus, flatten villi, and septa, which separate neighboring lacuna. And it's called cotyledon. It's lobule and structural unit of placenta. It's endometrium with spiral arteries. They carry blood here. It circulates here. It gives oxygen and receives carbon dioxide from embryo. And those substances, they pass through the wall of villi. And after that, venous blood goes to the venous blood vessels. And here we can see a placenta barrier, which provides gas exchange between the embryo and mother. All substances, they pass through this barrier. Oxygen goes from mother to embryo, carbon dioxide from embryo to mother, and also nutrients are going from mother to embryo, and waste products from embryo to mother. So spiral arteries, they are carrying here arterial blood, and arterial blood gives oxygen to blood of embryo, but in embryonic blood vessels they carry venous blood to the placenta, and venous blood without huge amounts of oxygen. So this venous blood, it receives oxygen, it releases carbon dioxide, and it becomes to be arterial. And mother's blood, after uptaking of carbon dioxide and after giving of oxygen, it becomes to be venous. Here we can see mother's blood, oxygenated blood, it comes to placenta, to cotyledon, and embryonic blood, which comes from embryo, it's venous. Venous blood comes from embryo, and it receives oxygen, releases carbon dioxide, and arterial blood goes to embryo from mother after this gas exchange. And mother's blood, after this exchange, it becomes to be venous, and deoxygenated blood from mother carries waste products from embryo to mother's circulative system. So here we can see that placenta and villi of chorion, they are site of gas exchange, and embryo's blood becomes to be arterial from venous, and mother's from arterial becomes to be venous. So here we can see that arterial blood, as a result of it, arterial blood goes back to embryo, and it provides functioning of embryo, because embryo is not breathing inside the uterus, so it receives all the oxygen from mother's blood. And here we can see two umbilical arteries. They are coming from embryo, and they carry venous blood from embryo. So this venous blood, it goes to placenta, to all the villi, and it's distributed in villi. And after the gas exchange, which is provided in the cotyledons, blood becomes to be arterial, and arterial blood is collected by the veins, and one placenta vein carries this blood away through the umbilical cord. It's umbilical vein, and it carries this blood to embryo. So here we can see opposite situation. Arteries, they carry blood from embryo, and this blood is venous, and vein, it carries blood to embryo, but this blood is arterial because it carries oxygenated blood after gas exchange in the placenta. And let's look at the structure of placenta. Also, we already know that placenta, it has cotyledons, and placenta, it has two sides. Its fetal side, where umbilical cord is attached, its fetal side and its umbilical cord. And also there is maternal side. It's attached to the endometrium. Here we can see endometrium and its maternal side of placenta. So two sides are present in placenta. Fetal side is covered with amnion. Later we'll talk about this. And its maternal side, it's turned to the endometrium, and it includes a functional layer of endometrium. And here we can see histological slide. So it's fetal side and it's maternal side. Here we can see fetal side, it's smooth because it's covered with amnion, amniotic epithelium, and the maternal side, it's not smooth. It contains a functional layer of endometrium of mother. And here we can see it's amniotic epithelium, and there are numerous villi in the fetal side. There are villi and mother's blood which fills the lacuna. And its maternal side of placenta, there are decidual cells. Here also glands, uterine glands are present here, they are bottoms, and villi. And also we can see here, we can find anchorin villus, which is attached to the endometrium. And also we can see here different villi. Its fetal side, it's covered with amnion. Under it there is extraembryonic mesoderm and numerous villi. And we can distinguish here cytotrophoblast and syncytiotrophoblast, which covers this villus. And inside this villus there is extraembryonic mesoderm with some Hofbauer cells, macrophages, and also there are blood vessels of embryo which carry here embryonic blood for gas exchange. And here we can see a barrier which separates fetal and mother's blood. And deeper, there is a maternal part of placenta. Here we can see numerous villi also, and also its endometrium functional layer with well-developed decidual cells. They are present here. They first of all provide feeding of embryo during histotrophic type of feeding, and also they produce hormone relaxin, which activates hyaluronidase in the pubic symphysis, and it relaxes pubic symphysis during the delivery, and it helps in the delivery because pubic symphysis becomes to be more mobile. And it's the structure of placenta, and also blood vessels are blood of mother. It's present here in lacuna, which is formed by the endometrium, and it's septum of the endometrium. And here we can see appearance of the villus, its earlier stage of the development. It's cytotrophoblast, it's syncytiotrophoblast, and it's extraembryonic mesoderm. So it's secondary villus, and some villi already they have blood vessels of embryo. So here we can find tertiary villi. And during the development of placenta, villi, they are aged, so they undergo the changes, dystrophic changes. So first of all, they are increased in size, and gradually cytotrophoblast, it disappears. It becomes to be very thin, and later it will disappear, and it will completely replaced by syncytiotrophoblast. And in some places this trophoblast completely disappears, and those places are covered by fibrinoid, special fibrin-like protein, which covers those places, empty places without trophoblast. Why do we need this fibrinoid? Because it covers the places with damaged trophoblast, and it protects villi from immune conflict with mother, and it keeps placenta barrier to be a barrier. And if this fibrinoid will not appear, the immune conflict between mother and child is possible. The fibrinoid of placenta may be present in fetal part. It's known as Langhans fibrinoid, which covers the villi. And also lacuna inside the endometrium, they are lined with fibrinoid of Rohr. Here we can see this fibrinoid is located in the maternal part of placenta. And here we can see uterus, its myometrium, its endometrium functional and basal layers, and its blastocyst is attached here, its adhesion during the implantation. After that it forms villi which provide invasion, and here we can see completion of implantation. So blastocyst is completely embedded in the functional layer. And here we can see that embryo and blastocyst, it develops not in the cavity of uterus, it develops in the wall of endometrium, namely in the functional layer of endometrium, or zona functionalis of endometrium, not in the cavity. It's located in the wall. But it's growing in the wall, so this wall goes inside the cavity, and here we can see next stage, and the entire development of embryo and then fetus, it's provided by the functional layer of endometrium, so it's involved in the formation of placenta. And it plays key role in the formation of placenta, and also it's involved in the formation of decidua. So placenta is formed as a result of fusion of cooperation of chorion and functional layer of endometrium. We already know that chorion, it includes cytotrophoblast, syncytotrophoblast, and extraembryonic mesoderm. It's mesoderm, cyto and syncytotrophoblast, they form chorion. And when chorion is implanted in the uterine wall, in the functional layer of endometrium, those two components, they form placenta. Chorion, it forms fetal part of placenta, fetal component, and functional layer of endometrium, it forms mother's component of placenta. So those two parts, those two structures, they are necessary for the formation of placenta. And entire development of embryo is present in the functional layer of endometrium. So here we can see that embryo goes in the functional layer after the implantation, and here we can see this development. And entire endometrium during pregnancy, it undergoes the changes, so it's not typical endometrium before the pregnancy. So functional layer, which usually is changed during menstrual cycles, it also has some changes during the pregnancy, and this endometrium modified during the pregnancy is called decidua. Decidua is changed functional layer of endometrium. Here we can see this decidua, and decidua because it's named because it will be separated from the endometrium during delivery. Here we can see this functional layer of endometrium. Basal layer, it's not involved in this, only functional layer, and changes, they lead to formation of decidua. And we can distinguish three parts of decidua. It's decidua basalis, where chorionic villi are growing inside the wall of uterus and where placenta develops. So decidua basalis, it's endometrium which is involved in the formation of placenta. It's decidua basalis. Decidua capsularis, here we can see that endometrium which covers the blastocyst and embryonic egg, it covers it and it forms capsule, so it's called decidua capsularis. And walls of uterus, they also are covered by functional layer of endometrium, and we call this decidua parietalis. And this part may be called also decidua marginalis, but usually it's described as a part of decidua parietalis. So here we can see decidua basalis, those part of functional of a layer of endometrium which participates in the formation of placenta. It's decidua capsularis and also decidua parietalis, and this part also may be called decidua marginalis. And also a very important embryonic organ, extraembryonic organ, it's amnion. And let's look at the amnion development. So we already know what is chorion. It is formed by trophoblast and extraembryonic mesoderm, and it provides formation of placenta, and chorion is covered with decidua capsularis. But inside this embryonic egg there is an amnion, amniotic cavity, which is filled with some fluid, and we usually know that embryo grows inside some vesicle, some bubble which is filled with fluid. So this vesicle or bubble, it's called amnion. And let's look at the amnion development, how this amnion will appear. So here we can see two vesicles, amniotic vesicle and yolk sac, which we already know. Here we can find them also, amniotic vesicle and its yolk sac. And it's amniotic vesicle, yolk sac, and amniotic vesicle, it grows faster than yolk sac, and it surrounds it from the lateral sides. Here we can see. And yolk sac goes inside, and its body fold, it will be umbilical cord, and it will be body of embryo. It will be body of embryo. It's embryonic ectoderm which covers the embryo. Embryonic endoderm goes inside the embryo. It will form digestive tube, and its yolk sac. It is rudimentary organ in human embryogenesis, and its amnion, which is formed by extraembryonic ectoderm. So extraembryonic ectoderm, it covers umbilical cord, developing umbilical cord, and also it forms the lining of amnion, amniotic cavity. So embryonic ectoderm, it covers the embryo, extraembryonic ectoderm, it covers umbilical cord and lines the amnion. And here we can see the source of amnion development. And here we can see embryo, it will be head, it will be bottom, and here we can see it's developing digestive tube, its umbilical cord, and its yolk duct, it will be yolk sac, and its amnion. And we know that embryo, it has mesoderm, it's third embryonic layer which fills the space between ectoderm and endoderm. It's embryonic mesoderm, it's mesoderm of the umbilical cord, which will be involved in the formation of umbilical cord. And also extraembryonic mesoderm appears, it covers the amnion, and also it will cover yolk sac. So here we can see this covering. And it's trophoblast which forms the villi, and it's extraembryonic mesoderm of the chorion. So chorion, it includes trophoblast and extraembryonic mesoderm, there are villi. And here you can see the space between amnion and chorion. This space, on this side there is extraembryonic mesoderm of amnion, and it's extraembryonic mesoderm of chorion. And its endometrium, functional layer of endometrium, it covers the chorion, it's decidua capsularis, it's decidua basalis, it's blood vessel of mother and lacuna with mother's blood. So villi of chorion, it's trophoblast with extraembryonic mesoderm, they are located here, and blood vessels from embryo, they are growing in the villi to receive the nutrients. So this place, it provides gas exchange, and it's developing placenta. And later these layers, they will be fused, and they will form common wall of sac or vesicle where embryo develops and fetus develops. So these layers, they will be fused, and embryo or fetus usually develops in some sac filled with fluid. So this sac, it's composed of epithelium of amnion, its extraembryonic ectoderm, extraembryonic mesoderm of amnion, extraembryonic mesoderm of chorion, its trophoblast, and functional layer of endometrium or decidua capsularis. It's sac which covers the embryo. And here we can see its amnion, and it will be fused with chorion. They will be fused, and they will form common covering. Its amnion, epithelium and extraembryonic mesoderm of amnion, its chorion, and also its decidua, which covers the embryo, its decidua capsularis. And here we can see its amnion, which lines the amniotic cavity, its epithelium of amnion, its embryonic ectoderm, and its extraembryonic ectoderm, its digestive tube, its yolk duct and yolk sac, its amnion or extraembryonic ectoderm, its extraembryonic mesoderm of amnion and chorion, which were fused, and its chorion, and also it's located inside the uterus in decidua, and decidua capsularis covers the chorion. So here we can see entire sac which covers the embryo, and then it will cover the fetus during the development. So it's the vesicle or bubble where embryo is located. And here we can see its decidua basalis, capsularis, and parietalis, and its uterine cavity. It's like letter C now, or sign of question. And later, this decidua capsularis will be fused with decidua parietalis on the late terms of pregnancy. And here we can see the fusion, and uterine cavity will be obliterated here, and it will be a small triangle in the lower part above the cervix of uterus till the late terms of pregnancy. So decidua capsularis will be fused with decidua parietalis. And here we can see this decidua. It includes changed endometrium, and also it includes different parts of extraembryonic organs, namely its chorion, which is growing inside the decidua, inside the functional layer of endometrium, and it forms placenta. So it's decidua of uterus on the late terms of pregnancy, and during the delivery, after childbirth, this decidua will be separated from the underlying basal layer of endometrium, and it will be pushed out from uterus. There are 3 periods of childbirth or of delivery. First period when uterine cervix is opened. Second, it's birth of baby. And third period, it is birth of extraembryonic organs. It's placenta and decidua, and they are pushed out from the uterus. So here we can see how placenta is separated from the wall of uterus, and also in other parts of the functional layer. Functional layer of endometrium is completely removed from uterus together with umbilical cord, and it's third period of delivery. And after this removal, here we can see after birth of embryo, entire decidua will be removed from uterus, and only basal layer of endometrium remains. So the situation is like process during the menstruation, when changed functional layer is removed each month. And it's removed, and basal layer, it provides regeneration of the functional layer. And the same situation happens after the delivery and during the delivery. So functional layer of the endometrium, this functional layer is removed, and only basal layer remains, and it provides the regeneration of the functional layer of endometrium. And also muscularis, during the pregnancy, it undergoes hypertrophy and hyperplasia. It's increased in volume and size. All cells, they increase their number and volume. and they play key role in the delivery. They push out baby, and also they push out placenta with decidua. And we know that there are 3 layers of myometrium: inner, middle, and outer. Middle layer, it contains big blood vessels, which give the branches for the endometrium. And after the removal of placenta and decidua, after removal of functional layer, there is a bleeding from the spiral arteries. And to prevent extra loss of blood, to prevent and decrease the bleeding, myometrium is contracted under influence of oxytocin, and it makes a compression on the blood vessels of the uterine wall, and they are compressed and bleeding is over. So if patient hasn't contraction of uterus, doctor should give her oxytocin. It will help to contract uterus after the delivery and to prevent bleeding. So here we can see uterus, and after the delivery, after the birth, regeneration will take place, as well as after the menstruation under influence of estrogens. So students, that's all. Thank you for your attention.